Hello, my name is Rebecca McPherson, also known as Joanna, and welcome to my poster presentation. I'm currently working towards a Masters of Science by Research, the working title of which is The Influence of Trophic Status and Environmental Factors upon Zooplankton Communities in Small Freshwater Lochs. Now, zooplankton are an important part of the ecology of small water bodies, as is illustrated by Figure 1. They can provide a unique view into these habitats. Small water bodies especially are highly sensitive to climate change, and by monitoring zooplankton, we can monitor changes in environment that we otherwise wouldn't know to look for. It is important, therefore, to establish a baseline from which to work. My project will work towards providing a preliminary look at community composition in relation to trophic status and establishing that baseline. For my location, I have chosen Scotland, which is full of small but relatively unstudied freshwater lochs. I will look at three types of water body: stock lochs, those with surrounding farm use; bird lochs, those with rookeries; and altitude lochs, those with no significant allocanthus inputs. I will use a multitude of methodologies enumerating bacteria and chlorophyll alpha, measuring water chemistry including nitrogen, phosphorus, ammonia, total organic carbon, and heavy metals, and finally identifying zooplankton using both morphological and metagenomic means. For the zooplankton I'll be looking at is rotifera and planktonic crustaceans, or copepods and cladocera. As my sampling is scheduled for August, a pilot study was established in April from which to hone my experimentation and identify flaws in my methodologies. We went to a farm site in Somerset and took samples from a well, a pump, and a pond. Chemical analysis showed that all water bodies were within expected norms and were not unduly polluted by the surrounding land use. Due to the unseasonally dry April, only the well could be sampled for zooplankton. Visible in Figure 5 is the crude well sample, which was teeming with copepods. From this sample, we identified Metacyclops planus, seen here in Figure 6, whose known ecology lined up with the habitat it was found in. There have been two other pilot studies planned, one in Norfolk and one in Hendon, in order to keep identifying and ironing out flaws in methodologies. For my August sampling, I expect to see differentiation in the communities from the three different types of loch, as well as a difference in trophy. Thank you very much for listening.